Okay, today as per our syllabus, we are going to do chapter 17, although we stopped at chapter 10 for midterm. Today we are going to do chapter 17 and then tomorrow we'll come back to chapter 11. So this is genetics of immunity or immunogenetics, and this is a content heavy lecture. There's a lot to understand. You know, if you go into higher biology, higher molecular biology, you'll realize that immunology is one of the most difficult parts of biological sciences. And then when you're combining genetics with immunology, it makes it really difficult, but it's also a very hot topic. So those who are looking for, you know, in future jobs in pharmaceuticals, in biotech industry, then immunogenetics is really important. So all of immune system, our immune system or lymphatic system, we call it, it's mainly based upon a single principle. Uh, our soldiers, our immune cells, they have to differentiate between what is foreign and what is self. So you have molecules, proteins inside your body, those are self. And your soldiers should be very tolerant, should be very friendly to the self antigens, something that the body recognizes. Those are self antigens. And then whatever your body does not have, okay, those are foreign things. And antigens means molecules which are usually protein, but they could be carbohydrates, this could be nucleic acids, anything, that's an antigen. So any antigen that your body has is a self antigen, and a smart, healthy body with a smart, healthy immune system recognizes self antigens and is very tolerant. And at the same time, that smart, healthy immune system is not tolerant towards foreign antigens. So anything that's coming from outside is an invader. It's a foreign antigen. It could be a bacteria, it could be a virus, it could be a fungal spore, it could be a protozoan, it could be, you know, a foreign cell coming into it. So that's a foreign antigen, and your immune system tries to get rid of it. And genetics, of course, plays a very important role in immunity. Okay, what kind of antibodies would be made? How strong will the antibodies be? How would the immune cells talk to each other? There are a lot of chemical messengers, okay, cytokines. And any mutation that occurs in those genes that are responsible for your immunity can lead to diseases or disorders like immunodeficiencies, autoimmune disorders where your own soldiers start attacking its own tissues, allergies, and even cancer. So the lymphatic system, which is known as the human immune system, we have 11 organ systems in our body, like digestive system, respiratory system, reproductive system. So similarly, we have lymphatic system, and lymphatic system is very similar to your circulatory or cardiovascular system. So just like in your circulatory system, you have blood circulating in the body, and there are blood vessels like arteries and veins. Similarly, in lymphatic system, we have a fluid circulating called lymph, very similar to blood plasma. And there are lymphatic vessels, and we have a network of these vessels all over our body. And lymph is what is flowing in these vessels. And the organs of lymphatic system include the spleen, the thymus, and the bone marrow inside your bones. So what is immunity? Immunity can be defined as your body's ability to fight or resist infections and diseases, okay? And in, you know, broadly, our body has two types of immunities. Innate, that means something that you're born with. You are, that's genetically predetermined. For example, all of us are born with our skin. We really don't have to do anything special to get a skin. And any invader that has to get inside your body has to travel through the skin. Or the fact that, you know, watering of the eyes, tears. So why does it happen? It happens because as soon as you get something into your eyes, it's automatically your eyes start watering. It's trying to wash it away. Whatever got into it, it could be dust, it could be debris, it could be a bug, whatever got into the eye. So that's also a form of protection. And you really didn't have to do anything special to get those tear glands and those tear ducts that would keep the eyes clear and safe. So that is innate immunity, while adaptive immunity is more specialized version of the immunity. This is also known as acquired immunity. This is something that you acquire after birth. As you get more and more exposure to different environmental triggers, your body develops this immunity. This is slower. Adaptive immunity is much slower than innate immunity, but of course it's more specific and way stronger, okay? So these are the levels of immune protection. You see here in the book that we are using here, it says the first level is, this is the first line of defense called the physical barriers. This is the second line of defense, where we have different types of proteins and different types of responses, like inflammatory response. We have fever, which is a systemic response. That means the entire body is responding against an attack. And then finally, the adaptive immunity. In a lot of other books, you will see that these two are pulled together, and both of these are included in innate immunity. But so innate immunity has actually two levels: first level, second level, and the third level of your body's immunity is the adaptive immunity. And adaptive immunity, as I just mentioned, is more specialized, it is slower, but it is stronger. And adaptive immunity is further divided into two types: one that is given by cells, called cellular immunity, and another that's given by your blood. So it's called humoral response. The fluid, any body fluid, is called humor. So humoral response. So the first line of defense, which is part of the innate immunity, are the physical barriers. What comes to your mind when you think of physical barriers? Our skin is like a brick wall. The cells of the skin are very close to each other. There is no space between them. So that is the first line of defense. Any mucus secretion, as soon as you breathe in air, along with the air, you're breathing in dust, you're breathing in fungal spores, you're breathing in bacteria. So there is protection inside your nose. There's a membrane that has mucus in it, okay, and it traps what I was trying to get inside. There's nasal hair. There are speed bumps on the side walls of your nose, okay. So all of those are physical barriers. Any tract inside the human body that is open to the outside, think of the respiratory tract, the digestive tract, for example, mouth, the urinary tract, the reproductive tract, any tract that is open to the outside will always have mucus inside it, so that if undesirable materials, whatever got inside, would be trapped by the mucus. It would not let the undesirable materials go inside, try to block it. So these are all non-specific, very generalized defense. So this is physical barriers. Moving to the second line of innate immunity. The second line of innate immunity is also not very specific, maybe a little bit more specific than the physical barriers, but still it is a very broad, generalized type of response, very fast, okay? Doesn't take long for your skin to act, doesn't take long for tears to come. And this immunity also involves inflammation. So anytime you cut yourself, let's say somewhere in your arm, the area around that cut, you might see a swelling there. That area might be hot to touch. There might be edema, that means swelling there. It's because a lot of fluid accumulates. All of these are actually symptoms of inflammatory response. This is your body's own immune system trying to fight off an infection. It is not caused by the cut itself. Yes, cut was the trigger, but it is actually your body's way of dealing with the emergency. And the inflammatory response actually helps the body to heal faster. Why? Because if there is pain there, you are less likely to move that area. So immobilization allows faster repair. Whenever there is more fluid means what? A lot of blood plasma leak there, and the more blood is taken to the spot of injury, the faster the repair, because blood is the main transport agency. So all of these, and then the cells from the blood, they come out, mainly the macrophages, which are like the California Highway Patrol. They are everywhere, okay? And anytime they see something undesirable, they're going to eat it up. So those are the macrophages, and their main job is phagocytosis. They eat up things that are undesirable. So all of these are part of inflammatory response. Inflammation, this is a localized inflammation, okay, in one area, because you cut yourself near the elbow, so that is where the inflammation is. The second line of defense involves a lot of different types of molecules, okay? Collectively, these are known as cytokines. And the cytokines could be interferons, these could be interleukins, this could be tumor necrosis factor. And you see how they are protecting you. Interferons are protecting you against viruses. Interleukins induce fever. You know, fever is a systemic inflammatory response. When a localized response doesn't help your body, then your body decides that, okay, the attack is way more serious and just a local response is not helping me. So let me just involve the whole of the body. So your white blood cells, it is your immune system that goes and tells the thermostat in your body. The thermostat is a small part in your brain called the hypothalamus. It goes and tells that thermostat, could you please increase the temperature for us, okay? So the regular body temperature is certain, and that temperature, the bacteria can thrive very well. But as soon as your hypothalamus resets the temperature, your body temperature starts going up. That's a fever. Who induced it? It's actually your immune system in response to the infection, of course. And as the temperature goes up, bacteria cannot survive there, but your immune cells can. Actually, your soldiers can fight better in that higher temperature. So it is inactivating the bacteria while your soldiers are working. And there's also another advantage. When your body temperatures are high, your liver and your spleen, they stop secreting some food for the bacteria. Okay, they secrete iron and zinc, which are actually food for the bacteria. Bacteria just love it. So when it's high temperature, your liver and zinc immediately get the message that temperature is high. That means bacterial infection is here. So I'm not going to release that iron and zinc. So the body is also trying to starve the enemy. So on one hand, the soldiers are being heated up, roasted bacteria, and they are being starved because no food supply from inside. So that is a fever. So that's why low-grade fever, actually, we should not take analgesics and try to bring the temperature down, because you are depriving your body of an opportunity to fight off the infection. But of course, fever needs to be monitored. And if your immune system gets over enthusiastic and the fever goes higher than 104 or 105 degrees Fahrenheit, then yes, you have to take medication to bring the temperature down. Interferons are protection against viruses, and actually the FDA uses some interferons against hepatitis C infection. It's a viral, I think it's interferon alpha that is being used. I may be wrong, it could be interferon gamma, but I forgot. Anyways, one of the interferons is being used to protect against viruses. The second line of defense also includes a group of 20 proteins called complement. These proteins work as a group, and they are usually, moving around in your blood circulation by themselves. And as soon, let's say, as soon as they see a bacteria, which is an enemy, and let's say one of the proteins saw it, so that protein rushes near that bacteria and starts calling its other 19 friends for help. So it's, as it is shouting, help, let's say 2 other complement proteins heard it, and they also rush towards the bacteria and they also start screaming. So now 3 of these complement proteins are screaming for help. So, like 4 more hear the screams, and they come and they also start screaming, kind of like lag. So it's like calling for help, and it goes in a cascade of events. It keeps increasing, the signal keeps increasing. And then all of the 20 complement proteins, they rush to that spot where the bacteria has been located. And what they do is they start making holes on the wall of the bacteria. Okay, so the bacteria has a wall around it, and the wall is being, there are pores being made, there are holes being made in the wall of the bacteria. How is that going to kill the bacteria? As the bacterial wall gets compromised, your interstitial fluid, that means the fluid inside your body, it rushes in. So the bacteria starts absorbing your fluid like a sponge, and then it explodes, it breaks, it lyses. So this is how the bacteria is being killed. But even those little parts, when the bacteria has been broken down, those little parts, they can still be toxic to your body. So your immune system doesn't stop there. Okay, it starts putting flags there, calling for your, signaling to your specialized soldiers, here there's an enemy here. Although I have broken it down into small pieces, but even these, I have dismantled the bacteria or the virus, but could you please come and check, make sure, confirm that these broken pieces are harmless, that they are not toxic. So this is where your more specialized immunity comes into action, which is adaptive immunity. So far, all of the generalized responses were innate immunity. Innate immunity is not specific. It cannot differentiate between, it may be able to differentiate between, okay, this is a bacteria, this is a virus, this is a fungus, but which kind of bacteria? Is it an, R bacteria, whatever S bacteria is, which kind of virus? Is it a retrovirus? Is it a DNA virus? It cannot do that, specialized differentiation. So this is where adaptive immunity comes in. Adaptive immunity is very, very specialized. This is not something that you're born with, you acquire it later on. This is very diverse because we have diverse types of enemies, right? So our soldiers also need to be diverse. They need to know how to fight different kinds of bacteria and viruses. It's extremely specific, which type of virus, okay? And most importantly, it has memory. So once an exposure or an encounter has occurred, your adaptive immunity remembers that exposure, that encounter. And the second time, if something similar comes and tries to attack your body, then it wouldn't take your body that long to make the soldiers. Usually what happens is the first time an exposure occurs, it takes about 2 weeks for your body to make antibodies, your blood to make antibodies, get those exact soldiers ready for that kind of a battle. But once a type of antibody has formed, then memory cells remain inside your body. And the second time the same bacteria comes in, then your memory cells will not take 2 weeks to make those antibodies again. 2 days antibodies are ready and fighting it out there, right? So there are two types of responses in adaptive immunity: humoral, which comes from the body fluid, and the cells that are present in your body fluid are known as B cells or B lymphocytes. And B cells do not fight directly. They are not like the army who are just going out there and fighting it out. They actually make proteins called antibodies. So antibodies are not cells. Okay, do not make the mistake of thinking that antibodies are cells. No, antibodies are simply proteins. They look like Y-shaped structures. So these are the Y-shaped proteins which are made by B cells, and these antibodies are the ones that are going to fight. Now, the second type of response is a cellular response or cell-mediated response. In cell-mediated response, there is another type of cells, another type of lymphocytes called T cells. And T cells are the ones who are the real soldiers. They go and they fight. It's like, face-to-face battle with the enemy. So the immune system, you see, has different types of soldiers, different types of responses being produced by the different types of cells that the immune system makes. So for example, all of your blood cells, everything, because all of your soldiers are your white blood cells, and all of your blood cells, actually they start their journey in the bone marrow, okay, in the red bone marrow. This is where all of your soldiers are made, in the bone marrow. Then from the bone marrow, the cells migrate through the blood circulation, okay? So they are, you see these blue arrows, they're in the blood circulation. Now some cells, some white blood cells called the monocytes, they move out of the blood system, and then they're known as macrophages, okay? This is the highway patrol that I was talking about. These are the cells that go in every part of your body, in the brain, in the skin, in your liver, in your digestive system. They're just there everywhere. And they eat up. Their job is to eat up undesirable things. They are called macrophages. And the other specialized cells, they will go, some cells will mature in the thymus, some cells will stay in your spleen. And these are the cells from the spleen, they go out and they become the B cells, okay, which will make the antibodies. While the T cells, which mature in the thymus, they will become mature T cells. And both these T cells and B cells together, they will give the adaptive immunity. The T cells will give you the cellular or the cell-mediated immunity, while the B cells will give you the humoral immunity, that means immunity by the antibodies. So how does this humoral immune response occur? Okay, so let's try to see in this figure here. There's an antigen that gets inside your body, a foreign antigen, a bacteria. Okay? Now, as soon as the bacteria is detected, what's going to happen? Those immunosurveillance cells, remember those cells which are present everywhere, the macrophages, they go and they immediately eat it up and they break it down into small pieces. Once they've broken it down into small pieces, they will take little parts of those pieces and stick it out of their surface. It's kind of showing to your specialized cells, I have something here, could you please come and take a look? Now there's a special type of T cell called helper T cell. This is a scout. So the helper T cell would go and would check. It's like, okay, let me check. Is this harmless or is it still harmful? If it is harmless, fine. Case ends there. If it is not, then the helper T cell would signal to the B cells and tell the B cells, there's a foreign antigen there and I need you to do something about it. So now the B cells, which was maybe only one number before, okay, this is just a hypothetical situation. Now the B cell says, okay, give me some time. Let me just increase myself in number. Let me proliferate. So this is proliferation. The B cell starts multiplying. Now there could be thousands of B cells and only, thousands of cells with different types of antibodies. And the B cell will first determine which type of antibody is needed. And only that B cell that can make that antibody will proliferate. Others will not proliferate, right? So B cells will proliferate, and when they proliferate, they become what is known as plasma cells, and some of them will become memory cells. The memory cells will be stored in the bank for future use. And it's the plasma cells that have these Y-shaped structures on their outer surface. And these Y-shaped structures are the antibodies. And the plasma cells will then release these antibodies, and the antibodies will go and they will attack that orange dot antigen. So they're going to take care of the toxic material. So you see here, this is the plasma cell. These are the antibodies. This was the bacterium that had gone in. Now, different kinds of plasma cells might be making different types of antibodies. So for one bacteria, the body has already activated, for example, 3 plasma cells here, because these 3 plasma cells make 3 different types of antibodies. And these 3 different types of antibodies recognize different parts of bacteria. So one antibody is going to attack from this direction, the other antibody is going to attack from this side, the third one comes from the back. So all these 3 different antibodies are trying to fight off this single bacteria. Now we are talking about antibodies. So what do antibodies look like? As I said, they are Y-shaped structures. They are proteins. And proteins are made up of chains of amino acids. What are chains of amino acids? We call them polypeptides. So if you see this structure here, this is one chain, this is another. So chain 1, chain 2, chain 3, and chain 4. There are 4 chains or 4 polypeptide chains. 2 of these are small. This one, number 1 and number 4 are small, while number 2 and number 3 are long. So there are 2 short chains, or we call them the light chains, and 2 long chains, we call them the heavy chains. And these chains are bound to each other, okay, by means of these chemical bonds. These are known as disulfide bonds, these SS in yellow. These are the disulfide bonds. Now each antibody has some region which is common to all antibodies. So all antibodies have this region which is known as a constant region. This is maintenance part. But every antibody also has a region that is different or unique in each antibody, because that region is required to fight a unique antigen. So this part, this outer part here, is the variable region, and the variable region is also known as the idiotype. The idiotype is the region which would be different in every single antibody. The constant region would be same. So this part is the antigen binding site. So as soon as you see a part of the bacteria, this is the antigen. This is where it binds, okay, the antigen binding site. This is the variable region or the idiotype. And the part of the antigen that is in contact with the antibody is known as the epitope. So this was a little bit of biology of the antibodies. What are the functions of antibodies? What exactly are they doing? The antibodies are simply using their strength in numbers. They're going there, they're binding to the antigen, the pathogen protein, or the whatever toxin is there, and is clumping it. It's grouping it all together and then inactivating it. It might precipitate. If you've done any chemistry, you will know how things precipitate down. They go down in a liquid. So it is clumping them, getting them pooled all together, bringing them down so as to not, letting them stay active, and then neutralizing them. And as they are clumping all of the antigens together, so they are making bigger and bigger clumps, right? And so then they are signaling those T cells, which are the soldiers that can fight face-to-face combats. It is signaling the T cells, here I have a lot of enemy soldiers pooled together. You could just come and take them all together instead of hunting for one by one, wherever they are hiding. So I have a lot of the enemy soldiers here, all down, kind of inactivated. You can come and kill them here. We have 5 major classes of antibodies. We call them immunoglobulins. So immunoglobulins, we write it in an abbreviated form as a capital I and small g. So immunoglobulins are antibodies. Class 1, first class is called IgA, IgD, IgE, IgG, and IgM. In short, remember MAG, M-A-D-G-E. So MAG are the 5 classes, and these are their functions. What is the function of each type of antibody? For example, in allergies, you see this one, okay, IgE. IgG is a type of antibody that a mother can give to a fetus when the baby is still inside the uterus. So this antibody diversity that we have, we have such a large population of different types of antibodies. This is caused by varying combinations, varying combinations of genes. Okay, how the genes arrange themselves, how they get spliced, how the RNA processing occurs. will lead to the formation of different types of antibodies. And the gene for antibodies, it has specific regions, okay? It has a V region called variable, the D diversity, and the G. So different combinations of this V, D, and J could create new versions of antibodies. So you see here, there could be V1, V2, V3, D1, D2, D3, J1, J2, J3, and depending on how, you remember from DNA to RNA, RNA to protein, there's this processing, splicing occurring. So what is the combination of these three regions would give rise to new type of antibody, or it would bring diversity in the antibodies. Then the next type of response, so far it was all humoral response or antibody mediated response. Now this one is cellular response or cell mediated response. This is where the T cells that actually fight face to face. Now T cells are very special type of lymphocytes. They're also your white blood cells. They start their journey as usual in the bone marrow, like all blood cells. Then from the bone marrow, they go into the blood circulation. Now they need some specific conditions, some special conditions to become mature. They need to go to an organ called thymus. The thymus is a small endocrine structure that lies above your heart. So it has to go there because it needs two hormones to become adults. If those hormones are not there, or if the thymus gland is not there, then your T cells, the person's T cells would never become adults. They would stay children, they would stay infants. And infant soldiers don't fight, right? We don't send our children into army or US Marines, and we don't send them to war. They are children. So similarly, if T cells remain immature, they will never be able to fight. And these are the most specialized soldiers. And one of the problems with aging is, as a person ages, is this thymus gland, it starts shrinking in size. So imagine it's like the location where your most specialized soldiers are going to become adults is getting smaller and smaller as a person gets older. So after 60 years of age, it starts shrinking. That's why elderly people, their immunity is not as strong as a young person's, because the thymus gland is getting smaller and is producing less hormones, and your T cells, the T cells are not getting a place to become adults. They stay immature. So the T cells which engage in direct fight, they have to be extremely smart. They should be able to differentiate between what is foreign and what is self. And that is why before they pass as soldiers from the thymus gland, they have to give an exam, okay? So when the T cell progenitors, they reach the thymus gland, and this is where the training is going to happen, the thymus gland would display all the types of self antigens that your body can have, okay? And the T cells should be able to recognize all of those self antigens. This is this, this is this, this is an antigen from the liver, this is an antigen from the stomach. If they make a mistake, and by mistake, if they think, oh, this is foreign, and if they cannot differentiate between it, that is the end of their story. The thymus will make sure that that T cell does never get the approval of going ahead. That T cell will be killed right there, okay? And the raw materials recycled to make new T cells. So the T cells that would bind to self antigens, that means those that attack self antigens, would get killed by apoptosis. Apoptosis means programmed cell death. This is a kind of cell suicide that your cell use to kill themselves if the cells are not doing their job. So T cells have to display immunological tolerance. They should be able to tolerate their own self antigens. And this is achieved by getting rid of, by killing those lymphocytes that are autoreactive, that means that attacked its own antigen. This is how you would avoid autoimmune disorders, because autoimmune disorders means a group of diseases where the person's soldiers, they cannot differentiate between self and foreign antigens, and they start attacking body's own tissues. So we have three types of T cells in our body, okay? The middle one, the cytotoxic T cells, these are known as the killer T cells. These are the real soldiers, the ones that fight. And helping these cytotoxic T cells are the helper T cells, which are the scouts. It is always the helper T cells that will be sent out first to assess the situation. And then the third category is the regulatory or suppressor T cells. These T cells are there to monitor the situation. Why? Because sometimes when the war has been going on for a long time and the cytotoxic T cells have been fighting, they lose track of what is going on. They just become over enthusiastic and keep killing and killing. So somebody has to be there standing in a neutral position, which is these suppressor T cells, who monitor the situation. And when they think that the enemy has been defeated and the T cells do not have to fight anymore, why waste energy? They signal to the cytotoxic T cells, hey, we've already won the war, stop, okay? No more fighting required. So these are the functions of these three types of T cells. So helper T cells, which are the scouts, they not only help the killer T cell, but they also help the B cells. So these are the scouts that are helping both humoral immunity and cellular immunity. Whenever there is a situation that needs to be assessed, a macrophage, presenting an antigen or something, it is the helper T cell that will go. And if it thinks that, yes, this situation needs adaptive immunity, it will signal to the B cell as well as the cytotoxic T cell. The B cell will go there, it will make antibodies. The killer T cell would go there and engage in face-to-face combat. And how do these cells talk to each other? How is the helper T cell going to secrete or send out a message? It's all chemical communication, right? And those chemical messengers are, again, the cytokines. We learned about cytokines, the interferons, the interleukin-2, and so on. So this table gives you types of cytokines and their function. How are they helping in immunity? Now this figure shows you how the killer T cells work. Remember that the killer T cells are the most specialized soldiers, and the worst or the most dreaded enemies of the human body are virus infected cells and cancer cells. So the cancer cells and the virus infected cells are taken care of by the cytotoxic T cells. If you see here, this pink cell, let's say, has become cancerous. That means it was a normal cell before, but now it has become immortal and abnormal, and this is a cancer cell. Now this cancer cell has been detected by a helper T cell, and the helper T cell has, it has already pointed out to the cytotoxic T cell that you have to come here and attack. So the cytotoxic T cell comes close to this, it attaches to the cancer T cell, okay, with means of a receptor. And then it starts secreting a particular molecule which is known as perforin. Perforin creates holes in the walls of the cancer cell. It creates perforations, just like the complement proteins kill the bacteria. So they make holes, and when the holes are being made, of course, fluid can rush inside and it can totally lyse the cancer cell. In addition to this is an attack from the outside. There's another mechanism that the killer T cells use, which is not depicted in the figure here, is that these killer T cells, they also have enzymes, pro-apoptotic enzymes, okay? Enzymes are proteins. Those pro-apoptotic proteins also, secretly sneak inside these cancer cells through these holes. They go inside, they go deep into the cancer cells, and then from inside the cancer cell, they tell the cancer cells, you're no good, kill yourself. Okay, so these are pro-apoptotic enzymes. That means enzymes that promote apoptosis. So these are telling the cancer cells. So imagine this two-pronged attack by your killer T cells. From the outside, perforin is making holes, and from deep inside, the enzymes have gone deep inside, and they're telling the cancer cells, you really need to commit suicide, you have to kill yourself. These are the various types of soldiers in your body, the various types of immune cells. There are the macrophages, right, the highway patrol, the mast cells, which are for allergies, the B cells, which make antibodies, the T cells, which are 3 types, then the neutrophils, which are like really small macrophages. They travel faster but eat smaller things. Now coming to the MHC, which stands for Major Histocompatibility Complex. This is a complex of about 70 genes, okay? And these genes are all located on the P arm, that means the short arm of chromosome number 6. And these genes all code for proteins that act as cell surface markers. That means they help in recognition. So how will your T cell recognize what is self and what is foreign? How would your antibodies recognize? Okay, this is because of markers which are sticking out from all cells. So we would have, our own cells would have different types of markers. A foreign cell would have different types of markers. So it is those markers, those proteins, that help us recognize, that help our immune system differentiate between self and non-self. So these protein surface markers, the MHC, okay, there are three functional groups of genes that make these proteins: class 1, class 2, and class 3. Class 1 and class 2 genes make special type of proteins called human leukocyte antigens, while class 3, they make plasma proteins which would carry out very non-specific functions. So these are more specific HLA, which help in our immune system recognizing. You know, you must have heard in organ transplant cases, why is it that we just cannot randomly transplant an organ from any donor into a recipient? Simply because there would be organ rejection. You cannot do that. You have to very carefully match the recipient and the donor. Why? Because the recipient's immune system is gonna totally reject a transplant, an organ from a donor if it does not match. So that is mainly carried out by the HLA. Now moving on to HLA. HLA, these genes make proteins. These proteins, they link with sugars and they form very specific glycoproteins. If you remember the chapter on cells, we had learned about glycoproteins which are sticking out from our cells. It is the glycoproteins that actually give us a lot of identity. You know, even dogs, when we say that the dogs can sniff smells and they can differentiate between people based on the smells, they are actually, glycoproteins have an important role there also, okay? So they give each of us unique markers. So the HLA glycoproteins can also recognize bacterial and viral proteins. So our proteins are very different from viral and bacterial proteins. And the processing of these antigens, you know, this ability to differentiate between this is human HLA antigen, this is viral antigen, this is known as antigen processing. This is a very complicated process in immunology. Now these HLA antigens that our body has, HLA genes make HLA proteins. So HLA proteins are called HLA antigens. We have different types of proteins. So we are calling them HLA class 1 antigens, HLA class 2 antigens. So HLA class 1 antigens, you can find them on all of your cell types. HLA class 2 are found only on antigen presenting cells. What are antigen presenting cells? Mostly your macrophages, those highway patrol, those immunosurveillance. So let's say this is one of your macrophage. This is the nucleus of this is part of your immune system. This is a bacterium that has just come in. So the macrophage rushes to eat it up. It eats it up. Breaks it down into small pieces. Now this is a little bit of the bacteria, and this little bit will be shown to your more specialized cells by the macrophage, because the macrophage cannot decide if this part is free to be secreted out. What if this part is also toxic? So somebody has to make sure that this is safe. So this is going to take it and show it off here. So this is presenting the antigen to your lymphocyte, because lymphocytes, the T cells, the B cells, are the more specialized ones. So this is called antigen presentation, okay? And this macrophage is now working as an antigen presenting cell. It's like, hey, I've already done the first part. I have broken down the bacteria, but the broken pieces are right here. Could you please come and check? So initially the helper T cell is going to come, the scout, assess the situation, and then if required, it's going to call for help, and the B cell and the cytotoxic or killer T cell would come and take care of it. So HLA diversity, we all vary in our immune responses, in our immune strength. This is because we are genetically also different. Of course, you know, health-wise, environmental influence also plays an important role. What is our lifestyle like? What kind of food we eat? So genes with multiple alleles determine your HLA type, okay? So, you know, what are the chances that two people would have the same HLA makeup? 1 in 10,000 unrelated people will share an HLA type by chance, okay? And for any transplant surgery to succeed, at least 4 major HLA genes have to be matched. Otherwise, the transplant surgery will not succeed. What happens when our immunity becomes defective, when it is abnormal? Now, immunity could be, there are some diseases where the immunity is defective because those were inherited. it was because of a genetic reason. Or it could be something that was later acquired. A virus attacked the body and totally killed the soldier cells. So now this person is immunocompromised. So this is an acquired one. This wouldn't be inherited. Different types of inherited immune deficiency. There are many types, okay, at least 20 types. So you see here, there are 2 examples here. I might have put more, so there are a lot more examples here. These are all genetic diseases, genetic problems with immune system. So immune deficiencies which were caused by mutations. For example, this one, SCID, severe combined immune deficiency. This impacts both humoral and cellular immunity. So this person would have no B cells or no T cells. An example is that of David Vetter. He was known as the Bumble Boy. He was born in 1971 in Houston, Texas. And his parents were both carriers of a recessive mutation. Their firstborn was a male child. He died in 15 days. And doctors had warned the parents that if they have another male child, then the chances of that child inheriting again and becoming homozygous for that recessive mutation is very high. Their second child was a daughter who was fine. And then their third child was again a boy, this one, but this time the parents were prepared. So as soon as the boy was born, they got him checked and he had also inherited this. It's an X-linked recessive disorder called SCID X1. He was born without a thymus gland, okay? So T cells cannot become mature. So he did not have his most trusted soldiers. There were no soldiers inside his body. That means even minor infection could kill him. So the doctors initially, when he was small, they put him, in incubators, very sterile environment, and then they created a vinyl bubble for the boy. And of course, as he grew older, then they had to make it bigger. So millions of, maybe about $2 million were spent to take care, and researchers were trying to find a solution for this child to survive. When he got older, then they tried to create another bubble at his home so that he could visit and stay at his home, because that was the entire world that he knew. Then NASA also, NASA created special, just like astronauts have space suits, they also created suits for this boy with a connection to the bubble so that no bacteria, no outside infection could reach him. His only hope was a bone marrow transplant, but they couldn't find a matching transplant. So finally, they decided to do a transplant from his sister. And what happened was the sister, she had a dormant virus in her bone marrow that was not detected in the pre-transplant screening because she was a healthy individual. So the virus couldn't affect her, and that was not detected, Epstein-Barr virus. But when the boy received the transplant from the sister, he developed cancer because the virus immediately, started acting, and he died within 15 days of the transplant. So that was a very sad story. Nowadays, genetic medicine has improved a lot, and this disease can be treated with genetic engineering, at least some forms. Then HIV and AIDS. HIV stands for human immunodeficiency virus. And this is a very dangerous virus because it attacks your immune system. So gets rid of it, infects your macrophages, it infects your helper T cells, okay? So with the macrophages gone and the helper T cells gone, it's like all of your immunosurveillance and your scout cells are gone. Nobody to protect you. So this is what a virus does. It comes first, it attaches to your, cell. This is a human cell, host cell. It leaves its protein code behind. It simply needs to get the RNA, the nucleic acid inside. This is the viral RNA. And then the viral RNA converts into DNA, and this DNA goes inside the nucleus, and then it binds to the host nucleus and modifies the DNA to make RNA. And this RNA will come out and make, instead of human proteins, it starts making viral proteins. So the virus has simply hijacked the host cell machinery, the host cell nutrients, the host cell everything for its own benefit. Okay, it starts creating its own viruses, and once the host cell is full of baby viruses, and the host cell just bursts, and all the baby viruses are released inside the body, and they start infecting more cells. This is how viruses in general, they attack. And this is what the HIV virus is more dangerous because, as I said, it attacks your immune cells instead of a cell somewhere else in the body. And with the helper T cell gone, Who's going to tell the B cells to make antibodies? Who's going to signal to the killer T cell to come and fight? Okay, so the virus would replicate very fast, and it keeps also mutating, changing. It can hide, and the viruses are very finicky. So once they infect a person, there is no guarantee that they're going to act immediately. Okay, the viruses can go and within 2 days it can just act and totally kill the person, or they can choose to remain dormant for days, weeks, months, or even years. Some viruses would just go inside and will not do anything for years. They'll just stay inactive. And then one fine day they wake up and like, no, I have to act now. So AIDS, which is caused by HIV, known as acquired immunodeficiency syndrome, this results from HIV infection. I think you guys know that just being HIV positive does not mean that the person has AIDS. HIV positive means the person has been infected by the HIV virus, but the person still doesn't have the disease. Okay, the HIV can just stay dormant, that means inactive for years, and not make the person sick. So that is just being HIV positive. And then when it starts acting up and starts attacking the immune system, that's when the person starts getting AIDS. So because of so much genetic diversity in the HIV, the treatment for AIDS would require different combinations of medications, okay? And there are drugs that inhibit different part of the viral life cycle. Some drugs could just stop the entry of virus into the T cells, the very beginning. Or once the virus has, let's say the virus has even gone inside, it would stop the virus from making more of its, changing its DNA, RNA, DNA, or even making of the viral protein. So different stages of the viral life cycle. These are some of the anti-HIV drugs that are in the market that are being used. This is what is the main. They could have different generic names. CCR5 gene. This is a very interesting gene because it codes for a protein which acts like a receptor on your cell membrane. And this is where the HIV virus likes to bind. So when the HIV virus attacks your cell, it binds to certain receptors. It's a co-receptor for HIV. Now, those individuals who get a mutation of this gene, so if they are homozygous for a particular deletion mutation, they actually are resistant to HIV infection because the HIV virus might enter their body, but they keep looking for the receptor to enter the cell. They cannot find the receptor. The receptor is not there. So these people are actually getting an evolutionary advantage. So although they had the mutation, but they have that advantage. And the individuals who are heterozygous, they might become infected, but still their chances, their prognosis is way better than another person who does not have this mutation, who's not heterozygous for this. Then we move to autoimmunity, where autoimmunity means your immune system attacks your own tissues, your own structures, okay? 5% of the population has an autoimmune disorder. It can arise in several ways. Your own T cells become dumb, become abnormal, and cannot differentiate between self and foreign, or viruses enter and somehow reprogram your T cells into attacking your own body cells. Here is a list of various autoimmune disorders. Some of the really common ones are diabetes mellitus. We all know, we all have some relative or some friend or a friend's friend who has diabetes. And this is, in this type of diabetes, the antibodies, they go and they attack your own pancreatic beta cells. These are the cells that make insulin, okay? And the antibodies, they think these beta cells are enemy, foreign. So they go and they kill those beta cells. With no beta cells in the pancreas, no insulin being made. With no insulin, nothing is there to control the sugar. So the sugar level starts shooting up. Another disease could be hemolytic anemia. Hemolytic anemia is where your own antibodies will kill the red blood cells. So of course the level of RBC goes down. Rheumatoid arthritis. This is where the antibodies would go and they would start eating up the cartilage, means that the tissue that is reducing the friction between two bones, okay? So it leads to arthritis, extremely painful in bones, in joints. Rh incompatibility. Rh incompatibility is related to antigens, okay, or proteins sticking out of your red blood cells. You know that we all have different blood groups. We have, some of us, we have type B blood. Some of us have type A blood. How are these blood types determined? These are determined by the kind of antigen that is simply sticking out of the red blood cell. So if a person has the A antigen sticking out, then this person's blood type becomes A. If a person has B antigens sticking out, then that person's blood type becomes B. If a person has both A and B sticking out, then that person's blood type becomes AB. While if a person does not have any antigen sticking out, then this person is like 0. So we call it a type O. So type O simply means absence of A or B. So whatever you have, your body loves it. That's a self antigen. And whatever you do not have, your body will make an antibody against it. So if you are blood type A, your body will make antibody against B. If you are blood type B, your body will make an antibody against A. If you are both A and B, will your body make an antibody against A or B? No, because you have both A and B, your body knows it. But if you are in blood type O, that means your body does not know A or B, neither. So then your body will make antibody against both A and B. So this is common for the blood group. Now there's a second layer of complication on this, which is called the Rh factor or the D antigen. So the D antigen can be present irrespective of whether you have A or B or AB or O. So either you have it, either you have the Rh antigen or you do not have the Rh antigen. So you can be an Rh positive or an Rh negative. So let's say you have antigen B in your blood and the Rh antigen. So then what is your blood group? Your blood type becomes B positive. If you have the antigen B, but you do not have the Rh factor, then your blood type is B negative. If you have A antigen, no Rh, you are A negative. If you have A antigen and Rh, you are A positive, and so on. So now the problem is when a woman gets pregnant with a fetus and they have different Rh factors. So the woman is, let's say, Rh negative. That means she does not have any Rh antigen. And because her partner was Rh positive, so the fetus has inherited the dad's blood group and the fetus is also Rh positive. Now what happens? The mom is Rh negative, the fetus is Rh positive. That means the fetus has the Rh or the D antigen. The mom doesn't have the D antigen. So for mom, that Rh antigen or that D antigen is foreign. So the mom, if it comes into contact with that Rh antigen, she will make antibodies against it. Those antibodies will go and they will kill the baby. They will kill the fetus. But usually this does not happen. Why? Because the maternal and the fetal circulation are kept separate. Okay. And usually whatever blood intermixing occurs, that occurs during the process of childbirth. And by that time, as the baby is, being born and some of the blood mixes, so the mother's blood starts making the antibody. It would take the mom 14 days to make the antibodies. By the time the baby is already out, so the first pregnancy is fine, safe, no problem. But then the mom's blood has received the first encounter with an Rh antigen. She now has made memory cells. So the second time she encounters this, it wouldn't take the mom 14 days to make those antibodies. Now let's say that this woman gets pregnant again. And because she is Rh negative, and let's say the second time also the fetus is Rh positive. So now the baby again is, the fetus is Rh positive and the mom is Rh negative and the mom has memory cells there. Now the second time, as the birthing process starts, as soon as the mom's blood gets into contact with the fetal blood circulation, immediately the antibodies will come and they will kill the fetus, right? So this is Rh incompatibility, and this disease is known as HDN, hemolytic disease of the newborn, HDN, or in other words, it is also called erythroblastosis fetalis. And the second baby would die if nothing is done. So nowadays, what happens is, as soon as a woman gets pregnant and the doctors, if they see that the woman is Rh negative, okay, and there's a chance that the woman's fetus would be Rh positive, they give the woman a particular shot, and that shot is known as RhoGAM. And in that shot, the shot prevents the buildup of the memory cells, those anti-Rh antibodies. So it makes the woman's body forget that it ever encountered a D antigen. So that means again, when the woman is pregnant the second time, the woman's body does not remember the previous encounter, the previous exposure. So this is how RhoGAM makes it safe for the second pregnancy. Now remember that, the first pregnancy is fine as long as there is no, internal bleeding anytime before childbirth. If the woman experiences, if there's any internal bleeding before childbirth, then the woman has to be given the ROGAM immediately. Or otherwise, the doctors would wait for a certain amount of time. There's a particular time when the ROGAM is given. But if there is any bleeding, then ROGAM is administered immediately. Allergies. Allergies are body's hyper reaction, hyper response to a non-threatening foreign substance, which is known as an allergen or a hapten. A hapten is something, it could be something as harmless as a little fleck of grass, a pollen or something. What happens is, if you hold a pen in your hand, let's say you have a red colored pen in your hand, and then you, on the other hand, you have a blue colored highlighter. You take the cap of the blue colored highlighter and you put it on top of your red colored pen. Okay, so that blue colored cap is the allergen. The allergen comes from outside. It is so small that it is harmless to the body. But the problem with the allergen is that it will come and sit on top of your own protein. So it is sitting on top of your red pen. And now your immune system, which otherwise recognizes the red pen as your own antigen, self-antigen, but it sees a blue cap on the red pen. So now this has become foreign protein. It has become enemy. So your immune system comes and attacks this red pen because it wants to destroy that little blue cap. Okay, it could have just ignored the little blue cap and nothing would have happened. But no, the immune system cannot do it because the blue cap is sitting on top of the red pen. It has become a big enemy now. So in order to destroy that little blue cap, your immune system will destroy that big red pen, which was your protein. So it is doing more harm to you than actually any benefit. So that is, in simple words, allergies. And that's why we try to have, take anti-allergies, because too much of an allergic reaction is very deleterious to the body. Different types of allergies. Allergies are very common these days. You see allergic rhinitis, which is main problem with the nose. You always have get a runny nose. You might get sneezes. Asthma is a type of allergy. Okay, it's a chronic disease. It's an inflammation of your airways, in the respiratory tract. It starts swelling. There's a lot more mucus secretion. So in allergic response also, your immune system gets activated. Okay, both humoral and cellular immunity are involved, and it is mainly IgE class of antibodies that are made. And these IgE class of antibodies, they go and they bind to very special cells called mast cells. And the mast cells would release mediators or other chemicals called histamine and heparin that produce a response against the invasion. So they would cause inflammation, they would cause that scratchy feeling inside in your eyes, the nose, rashes, and asthma. And a severe form of allergy, which could be really fatal, is known as anaphylaxis or the anaphylactic shock. Okay, this would totally choke your airways, causes death in the person. So a lot of anti-allergies, if you go over the counter, you see them, you see the antihistamines. Okay, these are a major class of anti-allergy drugs. Some of the more expensive ones, like Singulair, those are mast cell stabilizers. So you see the mast cell here, okay, and the mast cells which are stopped from releasing these. So those are also used in anti-allergy reactions. So this is a genetic pathway of how allergy sets in. For example, there's a particular mutation in this gene called filaggrin gene, and this starts an allergic reaction to something like a peanut allergy. If this is a healthy skin, you see the allergen coming in and not causing any problem. But if there's a mutation, then this allergen comes in and the skin does not provide a very efficient barrier, and the body's immune system is immediately activated, and you see all these different types of symptoms of allergies. Vaccination. Okay? Medical science has progressed so much. We have vaccinations against a lot of diseases. Some vaccinations you take once in a lifetime or maybe 2 or 3 doses, and they protect you forever, while some other vaccinations, you may need doses every year, like flu vaccines, because the virus keeps mutating. So you have to, so every year the CDC predicts, this year, these are the viruses that will come, become very active. So you need vaccines against those. So what are actually vaccines using? The vaccines simply use those foreign antigens. Okay, so let's say this vaccine is against this year's flu. So what will the vaccine use? It will take some flu virus and it would inactivate it or heat kill it. Okay, so they will make the virus very inactive, and that is what is given inside your body. So it is the same virus that you are getting, but the difference is that first of all, it's in very small amounts, and second of all, the virus has been inactivated, so it cannot really give you a full-blown disease. But the advantage of getting the shot is that as soon as those antigens are injected inside your body, your immune system is triggered. Okay, it's like the immune system immediately wakes up, because it would take 2 weeks for your immune system to make antibodies. So the immune system would immediately wake up and prepare antibodies. So that's why you take the flu shot before the flu season starts, so that your immune system is all active and it is ready with the antibodies that it needs to fight the infection. The first vaccine was created by this scientist called Edward Jenner. And in those days, smallpox was a big killer. Anybody who got it died or, would be debilitated for life, scarred for life. And he found out when, he was a physician, and he found out that women who were working with cows a lot, for milking and all that, they didn't get smallpox, or even if they did, it was kind of a milder form. So that set up a thought process as to what is going on with these women. Why, on an average, they don't get as badly affected as other people? And that made him realize maybe something was there that was saving them. And he realized that these women would often get infected with a milder version of a disease called cowpox. Okay, and then once they had the cowpox, they were acting as if they were immune to this killer disease, smallpox. So that is what made him think of, why don't I try this cowpox? And in those days, medical research laws were not that stringent and all. So he wanted to try this as a vaccine. He wanted to use some cowpox. And since there was no volunteer, he used his own son. And he injected him with cowpox. There was no guarantee. It might have killed his son, but he was lucky. His son survived, and his son did not get smallpox. And so this is how the story of vaccination started. Vaccines so far, still these days, they are mostly injections, but you might have seen nasal sprays that have, even the flu vaccine now has this nasal spray version. So these are simply, in whichever form you get the vaccine, these are stimulating your immune cells. Immunotherapy is also nowadays, even Loma Linda University here has a full division, that is devoted to immunotherapy, especially the development of the monoclonal antibodies. What they do is, let's say they take a mouse, and the antigen against which you need a medicine, you inject that antigen into the mouse. The mouse makes antibodies. So then you take the B cells from the mouse on which the antibodies are there, and then you grow these along with some cancer cells. Why? Because cancer cells are immortal. They just keep on living forever. Other cells are mortal cells. So you create a combination of these two. We call them hybridomas. And then you take the hybridomas and you separate, again, you separate the cancer cell out from them. You separate the harmful part out of it. And now what you have is an immortal, cell ready to fight, okay? And these have the antibodies. So those are known as monoclonal antibodies. Monoclonal antibodies have been used. You see two clinical applications here, Herceptin, which is against breast cancer, and of course the home pregnancy test strips that you use, which use a particular hormone. This is called human chorionic gonadotropin hormone. Transplantation can be so many types, okay? If it is within, let's say this is the person, and the person just receives from one part of the body to another, then that would be an autograft, okay? If the person has an identical twin, that's the best, because then between an identical twin, then it's an isograft. Between two unrelated person, then this is the most common one. This is called allograft. And if it's from a different species, then it's a xenograft. But for any transplantation to be successful, it has to be perfectly matched. So graft rejection is the most common problem, where the immune system can easily identify this as foreign and it just totally rejects it. And there are two types of rejection. First is a hyperacute rejection. Okay, it's a very severe form of graft rejection, while graft versus host disease, this occurs in bone marrow transplant. So, whenever people are given transplants, they are put on, immune suppressors. So your immune system is intentionally kept suppressed during that time so that your immune system does not act. So genomic view of immunity: in order for us to win the war against pathogens, we really have to sequence the pathogens. We need to know their genetic sequence in order for us to make things against them. Crowd diseases like COVID-19, we know these diseases spread very rapidly through unexposed populations. Bioweapons, okay, bacteria and viruses have been used several times in history as bioweapons. And we're done. Any questions? Okay, it's almost time for your discussion session. If you have not put your name in the chat box, please do so before you sign out. And I'll see you tomorrow with chapter 11.